Morning, guys. Well, we're starting chapter 11 today. Chapter 11 is about molecular genetics. In the last chapter, we learned about Gregor Mendel and all of the work that he did in helping us to have a better understanding of genetics. But realistically, when it comes down to it, genetics is the genes, or how we know where the genes are located is actually on the DNA. And so this section, or this chapter rather, is titled Molecular Genetics, because we're talking about how genetics works on a molecular level, not necessarily the alleles, but rather how that information gets passed from one generation to the next. So let's get started, chapter 11. 12.1 is about DNA, and more specifically about how we figured out what the genetic material is. We know, and we've talked about chromosomes, and we've talked about alleles, and the fact that some things get have different inheritance patterns that they follow, like incomplete dominance and codominance and epistasis, things like that. But now we're talking more specifically about what's happening in those molecules that make up the DNA. So we know about Gregor Mendel. We know Gregor Mendel was doing a lot of studies with pea plants and understanding how those things got traced through generations or got passed from the parental generations to the offspring. But now we're talking more specifically about how and why that stuff gets passed. So in the early 1900s, scientists are trying to figure out, now we have a better understanding of genetics and what it is, and we understand that it gets passed on from parent to parent, or from the parent generation to the children or the offspring. But what's not understood is what it actually is, what the molecule is that's responsible for creating heredity, or those things tracing from one generation to the next. So, excuse me. So they're looking for the molecule responsible for that. In 1928, then a scientist by the name of Griffith was starting to do studies with Streptococcus pneumoniae, and that's a type of bacteria that causes pneumonia. And so what he was doing, and if you look at this picture, it'll help a little bit, but what he was doing was he was taking the rough strain and the smooth strain and injecting mice with those strains of strep pneumonia, or the bacteria that causes pneumonia. And what he was finding was that, okay, so there's virulent and non-virulent, okay? So the virulent ones are the ones that are gonna cause more problems and cause death or disease. The non-virulent ones are the ones that don't, okay? So stick with me for just a second. Okay, so he takes the rough strain, injects the mouse lives just fine, no problems at all. Okay, the next group then, he takes the smooth strain, which is the virulent strain, injects the mouse, and the mouse dies. Okay, so then what he does is he takes that same strain, the virulent, the smooth strain, and he kills the bacteria with heat. Okay, so the bacteria, the structure and the bacteria is still there, but it's just not alive anymore. Okay, so what he does then is he takes the bacteria that's been killed and injects the mouse with that bacteria, and the mouse lives. Okay, so we heat killed the bacteria. That's kind of like how immunizations work. When people get immunized for stuff like for the flu or whatever it is, the influenza virus is in there, but it's been killed, right? So sometimes people will complain, oh, I get the shot and then I feel sick. Well, that's because your body is mounting an immune response to the bacteria, or to the virus rather, even though the virus is dead. Okay, so let me get back to the experiment here. All right, so rough strain, mouse was fine. Smooth strain, the mouse dies. Smooth strain, but the virus or the bacteria killed, and the mouse lives. Okay, now then he takes the rough strain, those are the ones that lived, and he mixes it with the heat-killed smooth strain, okay? So the heat-killed one did not have a problem for the mouse previously. Well, what happens then is he injects some live bacteria that don't cause problems with some dead bacteria that don't cause problems, right? So neither one of them is supposed to cause problems. And what ends up happening is that the mouse dies, okay? So we have live, okay bacteria, dead bacteria that does cause problems, inject the mouse and the mouse dies. We would have expected the mouse to live through that. So obviously now we're understanding that something's happening between those bacteria, and we're going to get to this in a minute, but what's happening is the bacteria are mutating so that they can get the DNA from the bad or the virulent bacteria and infect the mouse, and then the mouse dies, okay? So what he figured out was that the R strain didn't cause disease, the S strain did cause disease, and we know now why the mouse died, because obviously there was something going on there. There was some mixing of those bacteria, and they don't know what that is yet, but we'll get to it in a little bit, okay? But that did provide some evidence to another group of scientists, Hershey and Chase, 20-something years later. They were trying to find the evidence that DNA was the factor that was allowing the bacteria to change. Okay, the bacteria had to change somehow in order for it to be deadly. Okay, so what they did was they took a bacteriophage, which is a type of bacteria or a virus that attacks bacteria, and they started to figure out or running some experiments with that. Okay, now one of the more important things to note that is a bacteriophage is made up of both DNA and protein. Okay, viruses cannot replicate themselves. All right. So in order for a virus to reproduce, it needs to have a different type of a cell, whether that cell is a bacteria cell or a healthy animal cell or whatever it is. It has to have another cell in order to reproduce. Okay. So what they did then was they used some radioactive labeling techniques, and by doing that, they could trace the DNA from the virus, right? So the virus here, this is what a virus looks like, or what we think of, we're gonna do some radioactive labeling on different parts of the virus, and then that virus is going to infect the DNA or the bacteria cells, okay? So what they could do then is they can trace the DNA and the protein based on what's radioactively labeled, okay? Phosphorus is one of the things that does not get used in proteins. Sulfur is something that does not get used in DNA. So they radioactively labeled these two things, and then they allowed these viruses to infect bacteria. And what they went to see was which one had radioactive DNA, okay, or potentially the protein. Okay, so the bacteria was infected by the viruses. What happens is the virus attaches to the outside of the cell. You can see it there, and you can see it there. The DNA from inside the virus gets injected into the bacteria cell, and then viruses work the same way, whether it's plant cells, animal cells, bacteria, whatever. They inject their DNA into a good, healthy cell, and then it tricks that cell into creating more viruses, okay? So the infected bacteria then, after they do this with both the sulfur and the phosphorus, is they infect the bacteria, and then they separate the viruses from the bacteria, and they can do that because viruses don't weigh as much, so to speak, and so they can separate them relatively easily. What they figured out from that was that DNA was the radioactively labeled molecules that were then passed on from generation to generation, okay? So from doing these experiments, we now understand that DNA has to be the genetic information or the genetic material that passes things on from one generation to the next, because the proteins don't get sent from one generation to the next. It's the sulfur in the sulfur molecules in the virus that are getting passed on from one generation to the next. So that's the DNA or virus DNA. In the 1920s, there was also some scientists that were working on trying to have a better understanding of the structure of the molecules for DNA or the nucleotides. Those are the subunits or the base units each of these is a nucleotide, okay? So here's the nucleotide, and you notice that, you know, this would be like a long chain of nucleotides like we see right here. There's different types of them we'll get to in just a second. But the nucleotides, every one of them is made up of a 5-carbon sugar, okay? That's this right here. Ooh. That's this circular type molecule right here. These carbon atoms are all labeled. This one is labeled 5', this one is labeled 3'. That's going to be important in just a little bit. We'll talk about it. The nitrogen base, okay, and then a phosphorus group or a phosphate group. Every single one of them has that. Now, more specifically, each of those, there are different types. There's adenine, guanine, thymine, and cytosine in the DNA. And then if we're talking about RNA, RNA has what we call uracil. Uracil replaces one of those nitrogen bases. Okay, so here's the nitrogen bases: adenine, guanine, thymine, cytosine, and then uracil is only in RNA. That's the only place that we find it, okay? It will take the place of one of those when after transcription happens, which that's a different section, okay? So then in the 1950s, a scientist by the name of Erwin Chargaff was analyzing or trying to figure out how these nitrogen bases work out. And what he had stumbled across was the fact that there are equal amounts of adenine and thymine, okay? So the A's and the T's are equal to one another, okay? And also the G's, the guanines, are equal to the amount of cytosines. Now, this is within a species, right? So we're talking about humans, A's equal T's and G's equal C's. This brought him to this understanding that they must be equal. Like somehow A's are paired together with T's because every time we have an A, there's also a T that goes along with it. And with G's, there's a C. Okay, so adenines pair with thymines, guanines pair with cytosines. All right, they come in 2 different groups. They're called purines and pyrimidines. Not terribly important that you understand that part, okay? But with this, he comes up with what's called Chargaff's rule. And that means that each of those, the A's and the T's, come in equal parts. A's and T's are equal, G's and C's are equal. So if there's 100 adenines, there's got to be 100 thymines. If there's 10 billion guanines, there has to be 10 billion cytosines, okay? Understanding that now helps more scientists to better understand the structure of DNA. So they're using, so we have this group of scientists, Rosalind Franklin, Maurice Wilkins, Francis Crick, and James Watson, who are trying to figure out what the structure of DNA is. Now, they're kind of working together, but not really. They're a little bit competing with one another. And, you know, having big discoveries in science is kind of a big deal. There's a lot of competition. Even though scientists are supposed to want to be sharing information with one another, there still tends to be a lot of competition amongst different labs. Now, Maurice Wilkins was pretty much working on his own. Rosalind Franklin was pretty much working on her own. And then Watson and Crick were working together as a team, okay? So Rosalind Franklin was doing something a little bit differently than the other scientists were doing. She's a physicist, and she's trying to figure out the structure of DNA using a process called X-ray crystallography. Now, excuse me, we know that X-rays cause problems. They can create mutations in DNA and ultimately cause cancer. Well, Rosalind Franklin, pardon me, is doing X-ray crystallography, and she comes up with this famous photo 51. Now, it doesn't look like it to us. It's really hard for us to see this, but if you're a scientist or a physicist that is studying the structure of these molecules, this picture makes a lot of sense to them. So she creates this photo 51 by taking an X-ray of a DNA molecule. Okay, now the more dramatic story, as it turns out, well, we'll get to that in a second, actually. So by analyzing the photo, they determined that this is a helical structure. And what that means, a helix is basically like taking a ladder and twisting it. Okay, so you have a ladder, you know, with the rungs in the middle, and what they've later determined was that the rungs that are in the middle would be like the adenines and guanines, the thymines and the cytosines. But then they take the whole thing and they twist it. Okay, so they put a twist on it, and then it gives us what we call the double helix or the double helical structure. Okay, so Watson and Crick used Rosalind Franklin's Photo 51. They used information from Chargaff and Chargaff's rule, and they built a model of the double helix. And what they did was they took the two, put them together, and then gave it a twist, and it matched the structure that Rosalind Franklin was able to prove using X-ray crystallography. Now, one thing I would like to point out, every time there's an A, it's matched up with a T, and they work together like a lock and a key, okay? Like enzymes and proteins, they fit together perfectly. G's cannot pair with A's, it doesn't work, okay? It's rounded, and this one is a pointed end. Now, realistically, the structure is not like that, but this is just to show you and to prove a point. A's pair with T's, C's pair with G's, and that makes sense then when we think about Chargaff's rule, okay? And then when we look at the DNA structure, we have the nitrogen bases are paired up, or the base pairs, and then they put those together, and then they just give it a twist. And when I say they, I'm talking about living things. So again, the double helix is like a twisted ladder. There's pairs of cytosine and guanine, adenine and thymine, and they create or support the molecules, and they give it like the steps or the rungs. This kind of pairing is what we call complementary pairing, because they complement one another, okay? The adenine pairs with the thymine very nicely. Guanine with cytosine very nicely. Cytosine is not going to pair with adenine, and guanine is not going to pair with thymine, okay? Though they don't complement each other. So in talking about the orientation of the two strands earlier, we mentioned the molecules, or the atoms rather, and when maybe someday we'll learn in a chemistry class about naming carbon or labeling carbon atoms. So when you look at the picture here, here's the 5' carbon, there's the 3' carbon, okay? And you can see that there's another nitrogen base attached to it, okay? One's attached at the 3', one's attached at the 5', okay? They're arranged in order so that the 3' attaches to the 5', and on the opposite strand, okay, so these strands are anti-parallel, okay, meaning that one strand is going up and the other strand is going down, okay, if that maybe makes a little more sense, okay? One strand is going up, the other one is going down, depending on how they're oriented, the 3' and the 5', okay? Now, the Watson and Crick are the ones that got credit for discovering the structure of DNA, but the more dramatic or the juicy story is that they broke into Rosalind Franklin's laboratory and stole Photo 51, and by stealing Photo 51 and using their information from Chargaff, they were able to come up with the structure of DNA right before Rosalind Franklin and Maurice Wilkins. You know, so they broke into her lab, they stole her work. They were the first ones to announce their discovery of the structure of DNA. They're the ones that are given credit for it. And subsequently, they won a Nobel Prize. You know, in the science world, that's like all sorts of fame and fortune. And it was published in Nature magazine. Maurice Wilkins and Rosalind Franklin also published articles, but once Watson and Crick had published their article before, they're the ones that technically get the credit for it. Rosalind Franklin ended up dying of cancer because of all of her work with x-rays, because at the time they didn't know that x-rays were that damaging to your DNA. She ended up coming down with cancer, and she died of cancer. She was given awarded the Nobel Prize after she had already passed away. But it wasn't until then that she was given credit for helping discover the structure of DNA. This is Watson and Crick, and this is the model that they came up with to help better understand the structure. So when we think about things, you know, we're talking about DNA, obviously really, really small molecules. And when we think about chromosomes and how that stuff works, we basically take a human chromosome that anywhere from 51 to 245, or I'm sorry, 51 million to about 245 million base pairs, okay? Adenines, guanines, thymines, and cytosines paired up. So one chromosome is, you know, has tons and tons and tons of those base pairs. There are strands then that are smaller than that. They're about 140 million base pairs. And they would be roughly 5 centimeters long if they were wrapped out in a straight line. But DNA coils around little things called histones. Okay, here's our histones here. They're coiled around those histone cores. They're like these little proteins. And then those create what we call a nucleosome. So this right here, you can see I'm circling it with the cursor. That all together creates a nucleosome. And they make up chromatin fibers that supercoil, and they give chromosomes their shape. Okay, so big picture, we have the DNA molecule, the DNA molecules, and they go in these long chains, create a double helix. It wraps around these histone proteins and creates those nucleosomes, and they get condensed. So when we think about a chromosome that's in prophase, and we have decondensing chromosomes, this is what it looks like when it's decondensing, okay? It looks like this stuff here. It recondenses itself when it needs to go through mitosis or meiosis, okay? All right, so that's it for 12, or it's actually 11.1. We got a new book a couple of years ago, and so the chapters are a little bit different, but we're actually in chapter 11 here. I'll go ahead and pause the video, and you can continue on if you want to. If not, we'll take a break here.